Hi everyone. So in 22.5 we were looking at proto-oncogenes and oncogenes, and I mentioned that another problem that we can see with cancer is tumor suppressor genes, so issues with tumor suppressor genes. So we're going to talk about some genetic changes that will actually inactivate a tumor suppressor gene. So these are kind of our backup or safety net that prevents the cancer from getting worse or the cell from going through uncontrolled cell division. And if we have genetic changes that can inactivate this kind of stopgap, then we can actually have a real problem. And we can talk about the different ways in which this loss of tumor suppressor genes can lead to cancer. There's some vocabulary here as well. So the first real kind of study on this, or the first finding of a tumor suppressor gene, was in retinoblastoma, and this is a tumor suppressor gene that prevents a cancerous growth. And so this was the first one that was identified, and the retinoblastoma causes a tumor in the retina of the eye. And there are two different types of this type of cancer, one of which is inherited, and then it occurs very early on in life. And then we can have the non-inherited, which occurs later in life. And this is much less common than non-inherited. But there was a researcher, and this is Alfred Knudsen, who he proposed that it was a two-hit model. And basically what he was meaning is that retinoblastoma was not just from one mutation, it actually required two mutations, and that has since been confirmed. And so what he thought about this was that retinoblastoma would have to have two different mutations, and those people who inherited the form already have the one hit, and all they need is a second mutation for that cancer to occur. Now, people with the non-inherited form must have two hits. They have to have two things go wrong because they didn't inherit that mutation in the gene. And so this one occurs much later in life. Generally, we think of this as an accumulation of mutations over time, and this only happens rarely. And so what does this mean? What do we mean by this? So one of the recent studies about this with retinoblastoma was that there's a protein which can actually suppress the proliferation of cancer cells. And this protein then we think of as a tumor suppression gene, right? The gene of this. So there was one that is called RB, and it actually had the ability to regulate a transcription factor. And I'll show a figure in just a minute of E2F. E2F is actually what activates the gene for cell cycle progression, meaning that it's what tells the cell that it's okay to go ahead and go through cell division. This RB gene can actually regulate how much of this is being produced. And so, can regulate if this binds. And so what RB can do is it can bind to E2F, and when it does that, it inhibits its activity. And what that means is if it's inhibited, it means that cell division cannot go through. And so this is kind of a check on the system. So if it deems that it's being transcribed too much, we have too much E2F, and we have lots and lots of the cell division, then RB binds it and it slows that down. However, if we have a mutation that takes out this tumor suppressing gene, RB, then what happens is there is no brakes to pull, no way to stop it. E2F is free to continually tell the cell to go through cell division. So here's a picture of this. This is RB, and when it is actually attached to the E2F, it's inactive. If we get phosphorylation of that RB, then what happens is we can get dissociation, and this can go on and bind and actually go through transcription. And what we see is that the cancer-causing mutations that eliminate RB will allow this to occur continuously. We don't have any of this being bound and then released and bound and released. It's just continually attached. And that particular target gene then is in the active state or always on. And because this is the one that tells the cell to continue through cell division, it means that we would see cancer in this particular cell. So the second tumor suppressing gene that was discovered and really is estimated to make up about 50% of all cancers are associated with some defect in this particular gene is the P53. So the P53 is a little bit different than the retinoblastoma one that is actually just stopping, basically inhibiting cell division. P53 is actually looking to determine if it has already incurred DNA damage. And so it's really looking for mutations. And so if P53 finds that there is some sort of DNA damage, it can activate genes that will promote that DNA to be repaired, or it could activate genes that will stop cell division and repress genes that are required for cell division, so stop that cell from going through uncontrolled cell division. And it is also possible that it will activate genes that promote apoptosis or programmed cell death. So if the cell is too damaged, or the DNA within the cell is too damaged, p53 is actually kind of the one that says, all right, it's time to take it out of the lineup, and will actually cause the cell to go through cell death. So what is apoptosis? Apoptosis is how cells in our body, when they're damaged, can be taken out, gotten rid of. And so it's a process that involves the cell shrinkage. It also condenses the chromatin, and the DNA then will become degraded. And ultimately, we have programmed cell death. And it's facilitated by some proteases. So these are enzymes that can break down proteins. And the ones that do this are called caspases. And these are often something that we think of as what is actually executing the cell. So when these caspases are released, they actually will break down these proteins and break down the cell. And so in apoptosis, the cell is broken into very small vesicles and eventually phagocytized by cells in the immune system. So it's cleared by the immune system. So just a quick table to kind of give you some of the different ways that these tumor suppressor genes can work. So RB falls into those ones that negatively regulate cell division. So they basically stop cells from over dividing. And then we have the other group that is the genes that maintain genome integrity. P53 is the one I talked about, but probably most of you have heard of BRCA1 and BRCA2. And these ones are both ones that we see with breast cancer, that there's a mutation here that actually then, if you have a mutation, you don't have that kind of safety net of these tumor suppressing genes taking care of the problem. So the tumor suppressor genes that I talked about fall into those two categories, right? We have the negatively regulated cell division, and that was the one we saw with a retinoblastoma. And then we have the ones that maintain the genome integrity. Basically, they're looking for any kind of a problem, and they then will go through and help fix it. So we can have either genome maintenance, which is what p53 can do, right? It's cellular mechanisms that will either prevent mutations or prevent those mutated cells from dividing or surviving. So it will either prevent that, or if it can't prevent the actual mutation, it will stop that one from becoming cancerous. We can also get checkpoint proteins that will detect these genetic abnormalities and prevent cell division. P53 can do this as well. So when it sees DNA is damaged, it can stop cell division. And then lastly, it can get the DNA to be repaired with enzymes. And so if it's a very small mutation, there's a chance that it could actually be repaired, and then it would be able to go through cell division again. So I'm going to pause here and finish up this part on tumor suppressor genes in another short lecture. If you have any questions, please let me know.